Hey guys, this has Ben and Dustin again, another episode of How Stuff Works. Go for it.
Alright, good morning everyone. We're going to be talking about C60 Fullerenes today, how they're made, how they're functionalized, and how they're used. Now remember, as with all of our other episodes, this is going to be a dramatization, so don't try it at home.
The first method we'll go over for creating C60 Fullerenes, or buckyballs as they're called, is arc discharge.
For our discharge, you can set up like this one, with two graphite electrodes extending into a container. The next step is you hit it with 25 volts of electricity. We might need safety glasses for this one. Flip the switch. Oof, that was violent. As you can see, the graphite deposited on the inside of the container is an ash. Then, you can use a benzene solution to separate out the impurities, and you find yourself with a fullerene.
So right here, we have a buckyball. Now as you can see, these individual little cracks right here, they are actually carbon atoms, and these lines right through here are bonds in between each of the carbon atoms. Most of them are single bonds, but we also have some double bonds in the structure.
Alright everyone, and the second method we will go over today will be laser ablation for creating C60 fullerenes.
And laser ablation can be demonstrated by striking a graphite target inside of an oven with a laser, but not just any laser, a pulse double neodymium doped yttrium aluminum garnet laser. Now as you can see, the laser is ablating the target. And after the laser has ablated the target, it will raise it to 2700 degrees Kelvin. Now after it raises to 2700 degrees Kelvin and cools, it will form a fullerene.
Alright, so we have a fullerene. What can you do with it, you might ask? Well, first off, because of its hydrophobic nature, you can't just go put it into the human body.
So fullerenes dispersed in an aqueous solution of a solvent such as water tend to aggregate together. Now how do you fix that? You fix that by introducing functionalization to the fullerenes. Now whenever they're functionalized, they tend to move randomly in the solution as opposed to aggregating together. So you may be curious what you can functionalize the particles with. You can functionalize it with amino acids, carboxylic acid, hydroxyl groups, and amphiphiles. Now once you functionalize the C60 fullerenes, they can be attached to an antibody, antigen, or viral vector to target a diseased cell. Whenever they target the diseased cell, they can eliminate them. Now elimination is important because current chemotherapy treatments can have very destructive effects on the body, such as rather than simply eliminating the diseased cells, current chemotherapy kills everything.
But targeting alone isn't enough. Fullerene by itself would not have the necessary punching power to damage the diseased cell, much like what was that? Oh well. But if you were to load a drug onto the fullerene, it would impact the diseased cell more like this.
Straight in the schnoz, and let's see it again on the old instant replay. Up Sterling directly to the face.
So, when you have a properly functionalized fullerene that is able to target a diseased cell, it attacks the cell much like the little pink hat that is in this video. Now, as you will see, the functionalized fullerene is very potent.
Well, there you have it, the development and possible use of fullerene. We hope you have enjoyed this very exciting edition of How Stuff Works. Thank you for joining me, Dustin, and Ben, and we will see you next time.